Hi, my name is Eugenia Ampofo, and I'm a summer trainee in the Mayan lab at Mount Sinai. Today, I'm excited to present Rummagene x RummaGEO, which is crossing the gene sets from Rummagene with the gene sets from RummaGEO for novel hypothesis generation. Before getting into describing this project, I would like to provide some context. Many biomedical research publications contain supporting materials, which are typically large tables that often contain information about gene sets. The data in these supporting tables is not indexed by search engines, and as a result, it's not easily reusable to inform new studies. To address this issue, the Mayan lab, led by data science analyst Daniel Clark, developed Rummagene, a database that hosts about 750,000 gene sets extracted from supporting materials of about 140,000 articles after scanning 6 million articles available on PubMed Central with a developed bot. The Mayan lab also developed a search engine that can be used to query the collection of gene sets. Here is a picture of the site's landing and its URL. Similarly, RummaGEO, also developed by Mayan Lab, automates the extraction of gene sets from the NCBI Gene Expression Omnibus. Giacomo Marino, a bioinformatics software engineer in the Mayan Lab, led this project to identify groups of samples from RNA-seq studies deposited in Geo and available on ArchES4 to automatically compute differential expression signatures from this data. Currently, there are over 300,000 human and mouse gene sets served for search by the RummaGEO web-based app, similar to the way the Rummagene search engine is serving gene sets. One of the questions that stemmed after the creation of these two related tools was, could there be any interesting insights by crossing the gene sets from these two new resources? And from there, to answer the question, we created Rummagene x RummaGEO. First, I extracted the Rummagene and RummaGEO gene sets from their respective sites. Next, each Rummagene set was compared with all the RummaGEO sets. For the mouse RummaGEO sets, we harmonized the mouse genes before crossing. And for each Rummagene set, the top 10 intersecting sets that met the p-value threshold of 0.001 and minimum 5 overlapping genes based on the Fisher's exact test were kept. We also removed all intersecting sets from the same studies to avoid redundancy. The intersecting sets for the set pairs that met the aforementioned criteria were retained and entered into a new database. Once we had all the sets in the database, we adopted the code from the Rummagene and RummaGEO applications to create the new tool, Rummagene x RummaGEO. To better understand the content of the Rummagene x RummaGEO database, we further analyzed and visualized the top 100K intersecting sets. So far, over 600,000 Rummagene sets have been crossed with over 150,000 mouse RummaGEO sets and over 130,000 human RummaGEO sets, leading to over 9 million gene sets in the Rummagene x RummaGEO database. Here is a UMAP of the top 100,000 sets as determined by p-value from each crossing of Rummagene with the human and mouse RummaGEO sets. We see a lot of distinct clusters that separate the human RummaGEO and the mouse RummaGEO sets, but there are some clusters that equally share both overlapping sets. We also created a distribution of gene set sizes for the top 100K of both crossings, and they are both similarly distributed and have an average size of about 500 genes. We also plotted the frequency of genes appearing in Rummagene x RummaGEO gene sets compared to their PubMed citations. And as can be seen, a substantial amount of genes that are highly present in Rummagene x RummaGEO have low citations. This may be useful for pointing out important understudied genes. To examine the knowledge space covered by Rummagene x RummaGEO, we overlaid its top gene sets with the enricher gene sets. We can observe that many of the Rummagene x RummaGEO gene sets are disjointed from the enricher sets. This suggests that Rummagene x RummaGEO may illuminate new areas of biology that aren't currently covered. And to further explore the potential of Rummagene as RummaGEO, we examined some of the highly overlapping sets within these clusters and found that most of these sets demonstrated relatedness between studies. Here is an example of two such sets that have very high overlap. One set came from a study in Rummagene and the other from a study processed by RummaGEO. These two sets were extracted from two related studies that examined stem cell regulation, one in the context of breast cancer and memory development, and the other looked at human embryonic stem cells from females, focusing on the long non-encoding RNA X-inactive specific transcript, OS6 for short. And as you can see here, it has pretty low p-value with a substantially high overlap. We submitted the overlapping genes between those two sets to Enricher, and based on current literature, we know that the high overlap supports the known relationship between memory development and the development of breast cancer, and exists, which regulates the expression of genes on the X chromosome. Thus, Rummagene x RummaGEO can be utilized as a resource for identifying related studies based on gene set overlap and potentially deriving novel hypotheses from surprising insights. Next, we'll talk about the Rummagene as RummaGEO search engine. Here is a screenshot of the landing page, which will be available soon. It looks very similar to the Rummagene and RummaGEO with similar features, including gene set search that allows users to impute and query against the Rummagene as RummaGEO site, along with other features like PMC search that allows users to find papers along with geo studies and try to find related studies and potentially derive new hypotheses. And when a user enters a query for the gene set search, here's an example of what the result page looks like. It contains descriptions and links to the Rummagene and RummaGEO gene sets and the overlap with the overlapping sets, including the number of overlapping genes, the p-values, and odds ratio. In summary, Rummagene as RummaGEO has 9 million new annotated gene sets, and examining the top overlapping set pairs is very interesting and may point to new and unexpected insights. The Rummagene as RummaGEO search engine will also enable researchers to discover new biology potentially in the context of their own research. In the future, we plan to keep examining the top overlaps, deploy the site, and try to cross the gene sets from Harmonisome and Enricher with the Rummagene as RummaGEO gene set to find sets that highly overlap across 3 or more sources. Now, I would like to acknowledge and thank the Mayan lab members and my fellow summer trainees, Dr. Mayan and Ms. Jenkins, for making this amazing research program possible, and lab members Daniel, Giacomo, Stephanie, and Errol for providing me with much needed support and resources that aided me in this project. Thank you.